Hello, this is Dr. Panigrai. Welcome to my biology classes for NEET exam. Today, I will discuss about the cell-mediated immunity. As you all know that the cell-mediated immunity, it is the part of acquired immunity. The acquired immunity has two branches. One is the cell-mediated immunity and another is the humoral immunity. Humoral immunity also known as the antibody-mediated immunity because where the antibody directly involved to neutralize the antigen or to kill the pathogens. That's why these are known as the antibody-mediated immunity. But here, the total matter is different. Here, the cell directly attacked the pathogens of the antigen or infected cells and try to neutralize, try to kill, and try to lysis the cells. So cells directly involved in this fighting mechanism against the pathogen. That's why it is known as the cell-mediated immunity. In cell-mediated immunity, several cells are involved, but the very important cell is the thymus derivative cell or known as the T cells. These T cells actually very much important cell. This is the T cells has the very main role in the cell-mediated immunity. And besides T cells, other cells also involved like the macrophages and NK cells, natural killer cells, and killer cells. So here I may say the 4 number of cells are involved in cell-mediated immunity, but particularly T cells are involved in cell-mediated immunity. Cell-mediated immunity indicates that the T cells actually mainly involved. On the basis of presence of glycoprotein on the surface of T cells, the T cells again classified into 2, and the glycoprotein which present on the surface of T cells, these are known as the cluster determinant or the CD. Those T cells contains the cluster determinant type 4. It should be written as CD4 or CD plus 4, and the cell is known as CD4 cells, and those cells having the cluster determinant type 8 or CD8, or you may say CD plus 8. So on the basis of the cluster determinant, the T cells classified into here 2 types. One is the CD4, another is the CD8. The CD4 cells actually on the basis of their function, it is known as the T helper cells, and the CD4 cells and such cell on the basis of their function known as the T cytotoxic cell. So the several types of T cells are present, but here mainly 2 CD4 cell and CD8 cells involved in the cell-mediated immunity. The cells which have been the cluster determinant number 4 known as the T helper cell, and the cluster determinant 8 if present, then the T cells is known as the cytotoxic cell. The helper cells and the cytotoxic cell, this differentiate on the basis of their functions, and the CD4 number 8 and the CD4 number 4, these are differentiate on the basis of their molecular weight or the in chemical nature. So macrophage, in case of cell-mediated immunity, such macrophage act as the phagocytic cell after engulf the antigen. It represent the antigen to the other immune cells. That's why it acts as the antigen presenting cell. NK cells and K cells, they are directly involved to lysis the infected cells. The NK cells which secrete the chemicals, you know, this is known as the perforin. And the K cells, the surface of the K cells which contains the Fc receptor of immunoglobulin. globulin G, and it is responsible for antibody dependent cell mediated cytotoxicity. So first come to the T helper cells, how it kill the or how it involved in cell mediated immunity, how it involved for our defense systems. Suppose that this is the microorganisms, and such microorganisms having the antigens on the surface. This is the microorganism. Such microorganism actually enveloped by the macrophage by the process of endocytosis. So such microorganism, it is enveloped by this is the nucleus of macrophage. It enveloped the antigen. After taking inside such macrophage, it process inside it and present the antigen to the other immune cells. This is the part of or pieces of antigens, and the macrophage represent such antigen on the surface in association with MHC, major histocompatibility complex 2. If the antigen actually represent in association with MHC molecule 2, then it is recognized by the T helper cell. Suppose that this is the T helper cell. Such T helper cell recognize the antigen by its receptor. So T helper cell recognize the antigen if antigen is associated with MHC molecules. After the recognition of antigen in association with MHC molecule on the surface of antigen presenting cell or the macrophages, the T cells activate. Now the T cells become activate, and such T cells which forms so many cells or a clone of T helper cells. It may forms or it may divide itself repeatedly and forms so many cells. It also activates other macrophages. To come and engulf the other microorganisms. So helper cell, it helps to activate the other immune cells. That's why it is known as the helper cells. It also secretes the chemicals known as the lymphokines. It is one type of lymphocytes, so which secrete the chemicals is known as the lymphokines. by the secretion of such lymphokines, other immune cells like macrophages, other cytotoxic cells or other immune cells, they come to that reaction site. So this is the role of T helper cell in the process of the cell mediated immunity. So this is the role of T helper cell. Now come to the T cytotoxic cells. Here the total process is the same, but in cytotoxic cells, which actually activate by the viral infected cells. So first, suppose that a virus enter into the cells. Suppose that this is the virus. Such virus engulfed and processed by this is the nucleus. Such virus enter into the phagocytic cells by the process of phagocytosis. After the process inside it, such virus or viral antigen represented to the other immune cells. If the viral antigen represented to the other immune cells in association with MHC1 molecule, then it is recognized by cytotoxic cells. So, if the antigen is represented in association with MHC1 molecule, then this antigen, it is recognized by T cytotoxic cell by its receptor. So the TC cell, it recognizes the antigen if the antigen is represent on the surface of the antigen presenting cell in association with MHC class 1 molecules. At that time, the TC cells it activated. This is the activation of TC cells. The TC cells get activated and which become cytotoxic, which becomes cytotoxic. Because of the cytotoxic, the viral infected cells get lysis. Let me draw another figure here. The viral infected cells, suppose that this is the fragment of virus. This is the nucleus. Because of the cytotoxicity of cytotoxic cell TC cell, the viral infected cells get lysis, and which prevent the proliferation of virus inside the APC or antigen presenting cell or the viral infected cell. So here we see that the viral infected cell actually kill or actually lysis by the TC cells after the activation, and it is activated by the binding of its receptor to the MSC class 1 molecule in association with antigen. This is the receptor, and it also activate and proliferate so many cytotoxic cells. Clone of TC cells. This is the role of cell-mediated immunity this is the role of cytotoxic cell in cell-mediated immunity. Now come to the macrophages. I already told that the macrophages, these are the phagocytic cell and which actually act as the antigen presenting cell. The macrophages, the second is the macrophages. These macrophages actually act as the antigen presenting cell. These are the phagocytic cell which represent the antigen to the other immune cells after processing the antigen within it. That's why these are known as the antigen presenting cell. Now come to the NK cells. The NK cells actually it is known as the natural killer cell. Such natural killer cell is able to kill the viral infected cells or the tumor cells. The natural killer cells. These natural killer cells, they are able to kill the viral infected cells or the tumor cells. The viral infected cells or the tumor cells, actually it is suppose that this is the tumor cell, nucleus, tumor. Suppose that this is the tumor cell or this is the viral infected cell. The viral infected cell, it is recognized or it is distinguished because of the presence of high molecular weight glycoprotein on the surface of the viral infected cell. So high molecular weight of glycoproteins present on the surface of viral infected cell. Such tumor cell, it is recognized by the NK cell because of the receptor of NK cell which binds with the high molecular glycoprotein which present on the surface of the viral infected cell or the tumor cell. It is recognized. Then NK cell come near to the viral infected cell or the tumor cells and touch to the NK cell, come and touch the tumor cells or the viral infected cells, and the T cells secrete the chemicals known as the perforin. Because of the secretions of perforin, the tumor cells or viral infected cells becomes porous because of the secretion of perforin chemicals which make the pores on the surface of the viral infected cell or the tumor cells. That's why the tumor infected cells get lysis, which prevent the growth of virus inside the viral infected cell, or the tumorous cell, which prevent the tumor spreading, which prevent the spreading of tumor cells because of the NK cells. Now come to the K cells. The K cell is the last cell, which is known as the killer cell. The surface of killer cell contains the Fc receptor. Suppose that this is the this is the K cells. The K cell which contains the Fc receptor of immunoglobulin G. Suppose that the cell is present, viral infected cell. Here the viral infected cell is present. This is the viral infected cell. This is the nucleus. And viral infected cell, it is distinguished on the basis of presence of antigen on the surface of the viral infected cell. The immunoglobulin G. Now the immunoglobulin G, which forms the bridge in between the viral infected cell. This is the immunoglobulin G, which binds the one side which bind to the Fc receptor and another side which bind to the antigen of viral infected cell, antigen. And after the formation of such bridge, the cell becomes, the viral infected cell, now it becomes toxic, or the toxic effects works on the viral infected cells. That's why the cells automatically get lysis. And this process is known as the antibody dependent cell mediated immunity, antibody dependent cytotoxicity. This is known as the antibody dependent cell mediated cytotoxicity. So antibody dependent, so here is the antibody dependent cell mediated, this is the cell mediated cytotoxicity. So after the formation of bridge, the cell becomes toxic. That's why it gets lysis. So I hope you understand better the total process of the cell mediated immunity. If you like, click on my next video to get the more video like this. Thank you for clicking.